Welcome all. A very good morning to all of you in the ISFCP dialogue series under the aegis of IQAC, IIC, ISFCP, organized by ISF College of Pharmacy and Autonomous College, Moga, Punjab. Today's special guest and speaker, Dr. Dilpreet Singh, and his title of talk is Quality by Design in Product Development. We welcome Dr. Dilpreet. Please proceed further.
Thank you very much, Dr. Siddharth Mehan, for the introduction. So, I am hereby to present my talk on the quality by design in product development. So, so quality by design is basically a systematic, holistic, and proactive approach to pharmaceutical development. So, systematic means we have to define the independent variables. We have to screen out the independent variables that has the tremendous efforts to provide an excellent development approach to pharmaceutical product. So it's a holistic approach also. So the holistic approach means we have to criticize the what we call as the negative approach to provide a positive and proactive simulations to develop or optimize pharmaceutical drug products. So it begins with the predefined objective. We need to create a risk objective approach. We need to create the balanced approach in order to define the systematic task in the product development. So quality by design is basically a systematic task and a systematic approach to develop a pharmaceutical product in order to replace the OVAD and the OFAD, that is one variable at a time and one factor at a time. So OVAD and OVAD are the conventional approaches to develop a pharmaceutical product. So OVAD, basically we have to optimize one variable at a time, and avoid is we have to optimize the one factor at a time. But when we talk about the QBD is basically we have to simultaneously evaluate the every critical variables in order to analyze what are the understanding between the process and the product development and process control, and emphasizes the critical task between the critical response as well as the product response. So based on the sound science and quality risk management, we have to define the predefined objective. What is the goal of the formulation? What is the need of the stability of the formulation? What's the route of administration? What is the defined objective behind the rationale of the selection of excipient? What are the overall cost involved with the particular product? So these are the predefined objective we need to take our considerations in order to prepare a series of holistic approaches to define a product development series steps. So this QBD is also under the ICH Q8R2, the product development approach in order to systematically better understanding between a product and the process. So from now, QBD is one of the most important part of regulatory perspective. So we have to file it, learn it, and then we have to develop it in order to prepare a generic model for the business strategies. So from the developmental point of view, there are some major amendments during review process because QBD is a holistic approach. So there are some considerations which we define with the regulatory point of view in order to approve our process simulation and product simulation to a PDR, that is called product development report, to create a what we call as the ANDA to NDA connection task. And our developmental approach and our product formulation approach, it should be the reproducible in nature. That reproducibility is also provide a batch stability formulation revision, and this stability failure creates a down regulation in the overall product approval. So QBD is also replacing these pros and cons situations, and we have to create a shorter time for generic product approval in order to reduce the longer time in case of the conventional approaches. So we have to approve the product, which should be not be marketed. It should be also be changed with the post-approval marketing changes, and also to reduce the prior approval supplements. So how QBD it will improve the series of the steps which will need the assistance in order to provide the assurance of product quality for the patient. So that assurance will be created when we need to understand what are the product development steps and what are the process design steps in order to understand the QBD objectives to upregulate the task-oriented process. So QBD is a task-oriented process. When we learn QBD, we need to understand what are the process-oriented steps, what are the product-oriented steps, and we need to connect these steps in order to simulate better understanding of the product. So we need to monitor the tracking and trending of the product and process designs. If we need to monitor and tracking and trending the product design, we need to signify our mathematical model, which will need to understand that which mathematical model is suitably fit for our product development steps. Or for example, if we need a tablet formulation, so for a tablet formulation, we know that there are multiple excipients in the tablet formulation. There are the multiple type of disintegrants, diluents, flow promoters, and in order to understand that excipient, we need to understand which the best technique is able to define our product development steps. For example, this excipient, it should be compatible for the dry granulation process, it should be compatible for the wet granulation process, it should be compatible for what we call it the direct compression method. So in order to get the answer of these questions, we need to define that QBD that ensures that high level of product quality and the safety, and it also give an answer that which technique and which suitability step will provide you a better understanding of the product and the process. So it should be more efficiently, it should be regulatory oversight. So, we've already discussed that the regulatory oversight means we have to file a patent or we need to file an NDA or ANDA approval. So QBD will create a basically a network bridge between an NDA and the ANDA approval process in order to get our SUTAG, that is called scale up and the post approval changes. And these scale up and post approval changes will provide a what we call as the cost cutting approach in order to get our product much cheaper and much stable. So it is basically the efficient and cost saving for the industry also. It increases the efficiency of manufacturing process also. It decreases the minimization and it eliminate potential compliance action which will able to get the steps associated with the conventional approaches for product development. So this is the overview of QBD. We need to firstly define the QTTP, that is called Quality Target Product Profile. What are the target area which we need to define in our product? In order to define the QTPP, we need to understand the product design and the understanding. This product design it includes the CQA, that is called Critical Quality Attributes, CPP, that is called Critical Process Parameters, CMA, that is called Critical Material Attributes, CRA is the Critical Response Analysis, and finally is the optimization, that is basically the numerical optimization and the graphical optimization. These 4 to 5 steps it should be interconnected with respect to each other. Because if we want to achieve the CRA, we can't achieve with the CMA alone. We need to compile the CMA, CPP, CQA in order to get our CRA better and much cheaper as compared to the conventional approaches. So product design and understanding, it involves the product design and control strategy. This control strategy, it defines that SQM, that is called Strategic Quality Management. The SQM, it involves, we need to firstly define what is the goal of our formulation. In order to goal, we need to conceive the idea. Out in order to conceive, we get a realistic picture of that idea, and realistic picture it get to systematic development, and systematic development it creates the product life cycle that is called continuous improvement. So these further steps it should be connected with respect to each other. So QTPP, product design and understanding, product design and development, control strategy, and continuous improvement. This is one of the major tasks involved in the QBD process. In order to prepare a QBD task oriented process, firstly we have to initiate what is the compatibility between the excipients. For the compatibility between the excipient, we have to understand what is the QTPP of our profile. So the conventional approaches is been further replaced with the what we call as the advanced approaches, that is called QTPP, CMA, CRA, CQA, CSS, and the continuous improvement. So the continuous improvement it involves the product reproducibility, which is much higher in QBD as compared to the OVAD and OFAD approaches. So the product reproducibility is being defined as the RPN, that is called reproducibility, probability, and the decisive number, that is called RPN probability number, and should be much more than 99% accuracy as compared to the OVAD and OFAD, which having just 0.1% accuracy. So it having a 99% folding improvement process in the QBD as compared to the conventional approach. So the elements of QBD we already discussed. QTPP, that is called Quality Target Product Profile, defining critical quality attributes, CQAs, performing the risk assessment, and after performing the risk assessment, what are the risks associated with the product and process? For example, if we work on the RMG, that is called Rapid Mixture Generator. So RMG it involves multiple risk assessment. For example, like hardness. For example, like friability. For example, like compression force. For example, like compaction force. So the overall risk assessment between the compression and the compaction force will create a much more effect on the critical quality attributes and the quality target product. In order to perform the risk assessment, we need to link that attributes that we created with the risk assessment. That what are the risk approaches which particularly deal with either the raw material, either the process parameter, or either the CQAs. After performing the risk assessment, if we need to find out that our risk assessment is associated with the raw material, it is associated with the process parameter, or it is associated with the CQA, we need to design and implement a control strategy. A control strategy is brought by a decisive action, that is basically the strategic quality management. That control strategy is also, it should be highly harmonious in nature, it should be highly flexible in nature, it should be completely reversible in nature in order to reduce the waste as well as the time and efforts created by the inventor to produce the product. So that control strategy is being defined by the TQM, that is called total quality management. TQM is supported by the SQM, that is called strategic quality management, and SPM is supported by the QBT. So these three or four factors, they are totally connected with respect to each other. So if we need to create a product from scratch, we need to create a control strategy which should be uh, managed with the product life cycle. It should be managed with the continuous improvement, and it should be created with the risk assessment, defining the CQA and the QTPP process. So we already discussed what is the QTPP, the quality target product profile. QTPP is basically the set of elements that defines the product profile. So what target or what goal we need to achieve. For example, if we need to prepare a tablet formulation, so for a tablet formulation, what are the target is we need to create a best hardness with reduced friability, with optimum drug content, with the optimum content uniformity and the optimum flexibility in the oral delivery. So that QTPP is further supported by the RLD. It is basically the response latest design, and the response latest design, it applicable should be the regulatory guidelines. So that regulatory guideline is brought by the approval process, either the product is NDA or the product is ANDA. So we need to define the QTPP in the initial part of the drug development. After assigning the QTPP, we need to define the component that create a direct effect on the safety, efficacy, identity, purity, and the potency of a product. That efficacy, safety, and identity, and the purity and the potency of a drug product is been critically analyzed by the assay, content uniformity, and non-critically appraised by the appearance. So that create a high risk, low risk, and the medium risk factors. And the high risk factor should be reversible in nature in order to reduce in order to remove the risk associated with the drug product. For example, the assay is one of the parameter. The content uniformity is one of the parameter of a tablet formulation. These are the critical variables, and these critical variables create a direct effect on the safety and efficacy of a product. And if we want to remove that particular risk associated with the assay and the content uniformity, so we need to firstly define what are the CQA and the CRA of the product. It's called critical quality attributes and critical response analysis. For example, what is the optimum design space? For example, the optimum design space of a disintegrant is lie between 0.5 to 2%. After 2%, the assay it should be decreased, or the content humidity also could be decreased. So if we design a optimum space between critical variables, that is basically called as the disintegrant, we know that the design space is fall between the 0.5 to 2%, and this 0.5 to 2%, it creates an improvement in the assay and the content uniformity, and it should be not been having the direct effect on the appearance. So that QTPP, CQA, and CRAs, but there is a direct effect on the product profile also. So this is a fixed and variable components. So fixed element, it must be present, for example, the dosage form and the strength. Variable element, it may have a range of acceptable values, example the tablet weight and the assay profile. So QTPP component for the IR tablet, for example, we have taken the example of the IR tablet, is called immediate release tablet. So QTPP components like the dosage form, route of administration, strength, weight, pharmacokinetics of a profile, what is the absorption profile, what is the Cmax, Tmax, and the area under the curve of a drug product when we take a drug orally, what is the appearance of a product, what is the identity of the product. For example, if drug is thermolabile or thermosensitive, photolabile or photosensitive, heat labile or heat sensitive. So we need to take care of these factors while designing the product during the QDB QBD profile. What is the what the goal of assay? For example, from the Indian Pharmacopoeia of View, 80 to 120% assay is the required of a tablet formulation. So, what are the impurities associated with the drug product? What is the final content uniformity of 20 tablets? For example, if batch size is 20 tablets and the strip of size is 20 strip per tablet, so we need to define what is the content unity. It should be less than 5% as per the Indian Pharmacopoeia. What is the final friability profile of your tablet? What is the overall dissolution? Because in the Q2 or in the Q1 range, 80 to 120% of dissolution is mandatory to pass the in vitro in vivo correlation steps. So the IVC, we need to firstly clear the dissolution profile of a drug. It should be within the mandate range. And after passing the dissolution profile, we have to shift toward the in vitro in vivo correlation, that is between the dissolution and the pharmacokinetics. After clearing the IVIVC, we have to first shift it toward the bioequivalence. That is basically the 80 to 120% similarity in the Cmax, Tmax, and the AUC of your formulation. For example, the IR versus SR tablet or the IR versus CR tablet. So, what are the residue solvent associated with the drug product? So what is the score tablet? So the score tablet means what are the validation patches of your tablet? What are the pre-screened patches of your tablet? So it should be the variation between the two halves, that is basically the validation batch as well as the pre-screened batch, and the dissolution of a half tablet, that is basically the half batches, it is as well as the validation batch in order to screen out the final batch. So orally disintegrated tablet, we need to screen out the hardness, disintegration time, and the container closure. So we also know that the orally disintegration tablet. So dissolution is one of the predetermined step in the ODT. So dissolution is mandated by the disintegration. So we need to firstly analyze what is the disintegration time of a tablet, what is the hardness of your tablet, what are the container closures, and what are the packaging material associated with the ODT. And these variables are one of the most CQA and up-regulating CQTBP in the QBT development. For the extended release products, so for the extended release product, we know that we have to extend the release profile up to 24-hour period of a time to reduce the dosing frequency. In order to reduce the dosing frequency, we need to give into a critical condition or chronic condition, including that alcohol-induced dose dumping, as well as the patient suffering from the impaired kidney function, patient suffering from the impaired liver function, or the dose-dependent approaches where the non-linearity in the pharmacokinetic profile could be observed. So that consideration, that type of cases, we need to favor the extended release products. So critical quality attributes, this is one of the subset of the QPDP. So critical quality attributes are basically the attributes of a response. For example, if we need to formulate microemulsion, so for the microemulsion, we need to define the particle size, that is the globule size. We need to define that zeta potential, which defines the what we call as the coagulation. We need to define the entrapment efficacy. We need to define the drug loading. It is one of the most CQA and most predominant CQA in the nanoemulsion or the microemulsion type of the formulation. So it includes the critical parameter that could be likely to change. So that changes is being initiated by the variation in the raw materials and the processes involved. For example, if there are different type of industry and there are different types of the processes for the same drug product from the same raw material, it could be used. So these different processes, it could be changed the critical quality attributes of a product. So these critical quality attributes of a product, it should be monitored throughout the drug product development. And this drug product development, it ensure that our drug product, it should be remains within our safe and the effective levels in order to reduce the side effect as well as the dose inconsistencies. QTPP and CQAs we already discussed. So these QTPP and CQA, we need to create a very strong bridge, because this QTPP and CMA, the information, the flow of information, it should be transferable with respect to each other. So this flow of information should be not be revoked, it should be not be disturbed. So that factors we need to take in the considerations. So we need to define that, for example, one of the QTPP component of a IR tablet is the strength, and that strength, it creates a direct effect on the assay profile, because when we evaluate our formulation for hardness as well as the friability, so that the strength, it could be reduced, and it could reduce the dissolution, and it could be reduce the assay profile of a drug, because the diffusion step has been masked and drug would not be released in a predetermined period of a time. So that QTPP element and the CQ element it should be harmonious with respect to each other. So QTPP and the specifications. So that QTPP and the specification it creates a desired target for overall drug development process. That QTPP element it creates a specification range between a dosage form and strength and non specification range including the different sectors criteria with respect to the industry provocation. So that acceptance criteria, it will vary by the industry to industry. So that QTPP change should be harmonious in nature. It should be the regulatory compliant. It should be the defined motion that our developed product it should be acceptable to every industry. So that if we need to create a very strong acceptance criteria, we need to specify that our critical quality attributes is a specification of what we call as the overall sequential test. It should be reference to analytical procedure. You should be having predefined success criteria with respect to your selected quality, critical quality attributes, and your selected critical response analysis in order to understand what are the key reference products with respect to your predetermined analytical procedures. For example, if we need to create a new drug product which is not been established yet in the world, so we need to firstly create what are the analytical procedures that we need to understand, what we need to detect that particular drug concentration in the body fluids as well as in the plasma concentration. So that specification is also helpful when we firstly define the QTPP elements that should be specified with respect to dosage form, that should be specified with respect to your body, that specified with respect to your circadian rhythm, and that specified with respect to your chronopharmacokinetics, that basically a biological circadian rhythm that because our body structure, our body physiological system has been different with respect to different human bodies. So should our drug product it should be not being what we call as it should be not being varied with respect to different concentrations in our body system. So we need to establish a very strong set of criteria of a drug product which should be conformed to be considered acceptable for its intended use. So defining a QTPP doesn't mean that we need to settle all acceptance criteria. Our product specification it should be begun after QTPP because the CQA, CRA, and CMA is one of the most challenging steps in the QBD drug product. So QBD tools, we need to firstly assign that what is the risk assessment associated with the drug product. To identify the relative risk level at the beginning of a product, to prioritize the limited developmental resources, to document the decision-making process throughout the development, to assess the needs of additional studies for the scale-up as well as the technology transfer, to identify the appropriate specifications, to identify the appropriate critical process parameters and the manufacturing controls, and to decrease the variability of the CQA. This is one of the most important points in order to understand the risk assessment because the risk is being further been classified into three types: low risk, medium risk, or high risk. And these three type of risk it should be reversible with respect to each other. If the risk is irreversible, the overall batch is totally spoiled, and it is basically one of the most dumping task in the industry, pharmaceutical industry process. So, in order to reverse the risk associated with the development process, firstly we need to document what are the decision making steps towards the development. For example, the one of the risk factors associated with the drug product is the hardness. So one of the hardness we call the hardness is lie between 2 to 5 according to Indian Pharmacopoeia. If the hardness is lie between more than 5 or less than 2 in the immediate release product. So what are the decision making process in order to scale up our product and on we need to firstly clear the appropriate specifications. We need to reduce the critical process parameters as well as the manufacturing control in order to basically to set the value between the unified range according to the our need of defining the goal, what is the actual hardness value of the immediate release tablet. So risk assessment. So the risk assessment for formulation, it including the starting material properties, level of components, and the manufacturing process. For the formulation point of view, the starting material it includes the raw materials, the specified ranges, the product development, the series of steps, the manufacturing operation, the manufacturing controls. What are the QA parameters? What are the QC parameters? And these parameters have some levels. For example, the level is been subdivided into three types: zero level, it is the low level, one is basically the mid level, and plus one is the high level. So these are the called as the coded values, and these coded values it creates a down regulation in the manufacturing process. So we have to convert the coded value into an actual value, and this actual value is helpful for clearing the risk associated with the drug product. So the actual value, it defines the component and processes and the ratio of component to process. And this component and the ratio of component to a process, it defines the D value, that is called desirability value. And this desirability value is will further been helpful to create a process flow chart. And this flow chart, it creates a potential failure models and risk theory models which associated with your drug product. If we want to create a potential model for a particular process, for example, the model include the HACCP, that is called hazard analysis and critical control points, FACCP, that is called failure model and critical control point analysis, failure model and effect analysis, FAPA. That models, it creates a risk evaluation process, risk analysis process, and risk removal process according with the material properties as well as the manufacturing process. So some methodologies are available for risk assessment. And the model here are is called FMEA, failure model and effect analysis, failure model effects, and critically analysis. FMEA, it defines the product to process variation. FME, it defines only the product variation, and critical analysis, it defines the CQA variation. Hazard analysis and operability analysis is also basically for food products also, because the food product is basically undergoing the pasteurization, and the pasteurization temperature is being fluctuating from the high temperature to a cool temperature. So we need to analyze what are the CTP steps, because we need to refrigerate our food product. So what are the supporting statistical tools, including the ANOVA, including the what we call as the internally sterilized residues, externally sterilized residues, what we call the risk operations, normal plot of residues, half normal plot, Pareto charts, ANOVA, what we call as the transformation to a lambda value, that is called box cost plot. And these supporting statistical tools will help to evaluate what are the risks associated with the particular product. So the risk basically, that the acceptable range of a risk, basically it defines RPD, it is called what we call as the repetitive, processive, and the decisive factor according to your response. For example, the repetitive means that same experiment, it creates the same type of the result, and this same of type of the results creates a disharmony with the process and product development. The same type of result, it could be the negative results. For example, firstly, we have to prepare a formulation and the particle size is observed to be 1000 nanometer. If again we have to prepare a formulation, the particle size is observed to be 900 nanometer. The third step, we have to prepare a formulation and the particle size is observed to be 50 nanometer. So what is the variability associated with, because the low value it arises between 50 nanometer and the high value it arises between the 1000 nanometer, and this high variation, it creates a disharmony with the product and the process development. So your result, it should be reproducible in nature. Your result, it should be upregulating in the sensor, and your manufacturing control should be what we call as the regulatory compliant, in order to get your NDA and ANDA approval to a 99% accuracy level. So it is neither been always appropriate nor always necessary to use a formal risk procedure. So that for informal risk procedure, it be considered acceptable according to the ICH Q9. So the quality by design for ANDAs, an example of an immediate release tablet, for example, the acetyltryptan tablet is 20 mg, is basically a PCS class 2 compound. It creates a low solubility profile across the physiological pH range. It's also been available in different polymorph form, alpha, beta, and gamma form. Polymorph 3 is the most stable polymorph. And we have to prepare with the roller compaction process. That fourth one of the critical quality attributes, including the assay, content uniformity, dissolution, and degradation, according with your drug substance, according to your component variable, including the microcrystalline cellulose to lactose, according to your level, that is basically the low level, mid level, or high level, according to your talc concentration, that is called flow promoter, according to your disintegrant concentration, that is called magnesium stearate. So that basically the variable, it creates a direct effect or high risk effect to your process selected, including the blending and compression, roller compaction, milling, final blending, and the compression. So in this table, we have to understand that the content uniformity produces a high risk on the blending, roller compaction, and milling, as well as the compression, as well as the degradation. It creates a lower effect on the one of the these processing step of your tablet formulation. So this situation, it understand that content uniformity and distribution is one of the most starting point of the critical quality attribute that we need to take into the considerations in order to prepare your drug product better. So that justification, we need to assign the risk associated with the material component. We have to assign the risk associated with your, what we call as the product development steps. So that assigned risk that create a suboptimal uh, variation in the flowability and blending efficacy, and that create a direct effect on the vessel. For example, like we have to create a high risk effect on the content uniformity through a blending as well as a roller compaction, that the particle surface diameter and the cohesiveness of the drug substance that creates a highly adverse impact to your flowability if not blended properly, and if blended properly, it may create a compression index that is called Cu, and this compression index creates the overall flowability of your drug product. For example, the dissolution is one of the medium assigned risk according to your final formulation, and if we increase the blending up to a minimal range, that will create a decreased dissolution, because the disintegration is one of the impactful process by the formulation of granules, and it ultimately creates a negative effect on the dissolution products. So the third, fourth, and fifth factor in the QBD is the critical material attributes, critical process parameter, and the critical quality attributes. So CMA is one of the material attributes, that is basically the component of your drug product. CPP is the component of the processing parameters, and CQA is the required result of CMA and CPP. So that CMA and CPP should be harmonious with respect to each other. So the properties of the in-process material should be inert in nature. It should not having any pharmacological action to your body. It should also be reproducible in nature. It should not be incompatible with your drug product also. That CMA, it should be downstream according to your defined unit operation. For example, if we prepare a CMA according to your input variables, and that input variables prepared a first unit operation, and that unit operation, it creates a first level of your drug product that has the predefined CPP, and that predefined CPP is further been uh, motionized into output variables, and that output variables, it creates a second unit operation, and that second unit operation, and that second unit operation, it contains your high risk variables. That high risk variable, it includes the content uniformity, it includes the dissolution, it includes the high risk steps according to your drug product, and that particular CPP is been further been listed to your high CQA, and it form a stable product. So that's unit operation steps, that connectivity between your unit operation step, that connectivity between your CMA between unit 1 and unit 2, it is harmonious with respect to each other, so that the manufacturing process parameter, so that the critical process parameter, so that the critical material attributes, it should be finalized and should be screened better in order to get your product to a better quality. So critical material attributes, that material attributes we have to select it during the risk assessment, and that selected attributes, it form a direct effect to your product quality attribute, that is basically the hygroscopicity, particle size, residual solvents, process impurities, as well as the chemical stability. These five attributes, it create a direct effect on the CQ, that is basically the physical attributes like the size and the speciality, assay, content uniformity, and the drug release. For example, the hygroscopicity, it produces a low risk on the drug release. While the solid state form, either the drug has been compacted to a drug compression, drug has been compacted to a fat granulation, drug has been compacted to a dry granulation process, it creates direct effect on the drug release. And the particle size is one of the most upregulating parameter, is one of the most strategic parameter, because we need to control the size within the formulation or after the formulation. So the particle size should be not be changed during the drug intake, or during the what we call the packing of a drug product. So the particle size, it creates a high risk effect on drug release. Small particle size, it creates a larger surface area which promote dissolution, and high particle size, it creates lower surface area and it creates low dissolution. So CVP, it identifies the high risk step according to your unit operation that affects the critical quality attributes. One of the process steps including the blending compression to roller compaction to milling to final blending, lubrication, and then the compress to form final tablet. And that one of the parameters that directly create a harmony effect, directly create a TDS effect on your product quality, critical quality attribute, that is basically the assay, content uniformity, dissolution, and the degradation. And that degradation and that assay content uniformity and the dissolution, it been further justified by the compression forces, by the dwelling forces, by the press speed, that basically creates a high risk assessment to your product. So QBD is one of the most what we call as the versatile technique that also create a justification that if the risk assessment is high between your main compression force and the dwelling space, that QBD is also provide a logical statement that why it creates a high risk profile with your assay, why it creates a high risk profile with your dissolution, why it creates a high risk profile with respect to your hardness. So on the basis of this table, we have to justify that risk assessment. For example, the compression force. Too high compression, it creates a suboptimal concentration. Too low compression, it creates a friable tablet. So we need to prepare the optimal compression force in order to get your tablet within the specified range of hardness as well as the friability. And ultimately, we get a perfect solution. So, on the basis of the dwelling speed, we need to firstly create the high contact uniformity according to your mixing time. That too high mixing time, it creates a what we call as the instant drug release, and that instant drug release, it negatively affects the dwelling speed. Should be not be too much minimum, but it should be the minimum enough to create a dissolution to your specified range, that is basically 90 to 120% according to the specified parameters or according to the United States pharmacopoeia level. So control strategy is one of the most predefined strategies associated with the drug product according to the process parameters as well as the product quality. That control strategy, it includes the input material attributes, equipment operating condition, in process control, finished product specifications, controls, and method and the frequency of monitoring and the controls. So firstly, we have to define that what is the control strategy according to your immediate release tablet. For example, we have to firstly channelize our formulation and the product understanding. Firstly, we have to create a risk assessment according to your formulation and process understanding, that is basically your CQA, CRA, and CMA. That risk assessment, it creates your defined CQA, CPP, and CMA. According to your defined CQA, CPP, and CMA, we have to create a acceptance criteria that what limit we need to get your specified product ranges into an optimal range. And we need to define a control strategy. It should be reversible, it should be the harmonious, it should be the regulatory compliant, it should be the flexible, it should have the unproduct testing ability, and it should be in a well-defined attribute range. So that traditional approach, is under the level 3, that is called danger zone, that is called AVAD, 2 AVAD, that creates a down regulation in the end product testing. It should be tightly constrained because we have not been defined any CQA, we not even define any CRA, and we do not define any CMA to a product. So we have to shift it to what the level 3 to level 1. So we have to create an enhanced approach, that is called real-time monitoring, that is called process analytical technology PAT, which is one of the most important parameter in the control strategy. And these PAT operations, it should be a real-time automatic control. It should be the flexible process enough to create your up-regulation task in your product and the process development approach. So one of the most important tools of QBD is DOE, that is called design of experiments. DOE is one of the most useful for the screening of variables with significant effect on your critical quality attributes. This is one of the classical approach which releases OVAT. It is having the limited number of experiments and it gives precise information. DOE helps study to get your interaction between a multiple factor at a time, which replace one factor at a time. It is used in the optimization studies, and it enables the creation of a design space in order to get your product within the specified range of a time. So design space is proposed by the applicant and is subjected to a regulatory assessment and approval. We already defined that if we need an optimum hardness, we need to define the concentration of disintegrant from the low level to high level. It is called 0.5 to 2% in the design space. So that design space, It will need by the operating space, characterization space, and the goal space. And the goal space is basically further be shifted towards your lab and pilot scale that can be proposed for the commercial development, and it should be verified at the production scale for your better development of your drug product. So one of the most demanding tasks in the control strategy is that it is called process analytical technology. It's basically versatile technique and a multi-dimensional approach for analyzing the critical quality and the process attributes at a single time. So if we need to analyze the critical attributes and the process attributes, we need to firstly analyze what are the raw and the in-process materials, and that inline or online or outline processes is been further been confounded by your process understanding, and that process understanding has been facilitated by your process analytical technology that creates opportunities for improvement, that and opportunities for improvement is been further been certified by the reduction in your risk process. It should be more reliable and a consistent process as compared to your VAT or FAT. It should be flexible enough because I have already discussed that your control strategy it should be flexible as well as totally reversible in nature. So we have to remove the risk easily, and our continual improvement should be not being disturbed. So we have basically it's a faster and better option to define the quality management system for your drug product, and we basically the opportunities with respect to your process enhancement capacities is also available with the PAT. So if we need to apply PAT in tablet manufacturing, so the different tablet manufacturing, it including the dispensing, wet granulation, drying, blending, and compression, and the technique involved for that particular PAT techniques, that is basically the NIR, near infrared, what we call as the Raman spectroscopies, strain gauge techniques, and the measurement it includes the identification of raw materials, moisture distribution, moisture content, what we call as the blend uniformity, compression force, and the content uniformity. And these stages of a process control and these stages of a response analysis is been brought by the what we call as the technique used. And these three connection has been created by the process analytical technology, which create a single step process to analyze your stage process, your technique process, and your measurement process at a one multiple factor at a single time. Is one of the best example. It is basically the near spectral probe NIR analyzer, which basically is installed on the basically the fluid bed dry granulation process FBD. So this FBD it creates what is called as the continuous monitoring of moisture level with your moisture sensitivity drugs to create your formulation in a stable task because one of the most stage taskman is the moisture level. The technique is the NIR analyzer, and the measurement technique is the moisture sensitivity because the drug is moisture sensitive. So this NIR analyzer, according to your TAT technique, is will continue to monitor that GLAD FBT, that is called fluid bed granulation technique, and it continuously monitor the amount of moisture available in your formulation during that dry compression process, and that dry compression process has been sub-modified by your modification process, that is basically with drying or without drying. So one of the second example is basically the polychromic spectral blender probe that working on the principle of RMG. It is called real mixture granulator, real-time blending uniformity. This is by what is called as the true process. It is called extruder analyzer. ठीक and that extruder analyzer has been brought by the mixing, followed by the blending, followed by the compressing of his excipient to get your duff into a optimal range, and that duff has been further been screened out by the roller compactor, and that roller compactor has been compacted your tablet to a versatile properties according to your shape and the size of capsule. So these steps, including the multiple task process, because the rolling, blending, and mixing has been suboptimal, has been sub-optimized by the real-time blending uniformity approach that is called polychromics spectral blender probe. So this is one of the most challenging question now in the pharmaceutical industry, that QBD is required or optional. So if we create a up-regulating feature in your quality target product profile, critical quality attributes, if we need to understand the product design and the development approach, we need to understand the critical process parameter and the critical control strategy. QBD is one of the most prime challenges in the pharmaceutical development. If we need to define only and only design space, process analytical technology, the QBD is optional. So this is one of the bridge that created between different QBD processes in the pharmaceutical industry and has been getting supported by the different variety of factors, including the CDP, CQA, CMA, CRA, and what we call as the CPP of your parameter, and that create a bridge between your what we call as the understanding and what is the travel capacity from the starting step to your final goal step, and that final goal step has been created by your QBD process, and this QBD process is the multi-dimensional. This is the one of the references for QBD, and I recommend everyone to get, if we learn to QBD for the product development. So there are some ICH guidelines that available information on the QBD for product development, including the Q8 R2, guidance for industry Q9 quality risk management, guidance for industry Q10 pharmaceutical quality system, guidance for industry PAT, quality by system for ANDAs, quality by system for ANDAs for the immediate release dosage form as well as the modified dosage form. And we have to prepare a QBR, it is called quality by design, but they are what we call rugged report. Basically, it has been updated by the yearly wise. So, in order to better understand what is the optimum requirement of a QBD for product development, these are the references suggested. So thank you very much for your time and consideration. I'm highly thankful to the chairman of our ISS College of Pharmacy, Shri Prabinder ji. I'm thankful to the director, the principal of the college, Professor Dr. G.D. Gupta sir. I'm thankful to Professor Dr. R.K. Naran, Vice Principal of the college. I'm thankful to IQAC coordinator of the college, the Professor Dr. Siddharth Nancy, for providing the opportunity for the recording of the lectures to get our country to a knowledgeable task. Thank you very much, sir.
Thank you, Dr. Dilpreet, for your kind information regarding the QBD analysis and its significance in pharmaceutical sectors. And we are very thankful to our all listeners. Please visit our website www.isfcp.org and our official YouTube channel ISFCP College of Pharmacy. Thank you, Dr. Dilpreet, and thank you all listeners. Thank you so much.
Thank you very much, sir.